Hello, my name is Paint, number 14, and today I have to summarize about chapter 4 and chapter 5. So first we're going to start about chromosomes and DNA. So, chromosomes we have in bacteria, which is having a circular DNA, but in the eukaryotes they're going to have the linear DNA. So, chromatin is the complex of DNA and proteins found in the nucleus of eukaryotic cells. And this is the process of the DNA into chromatins. So first DNA, they're gonna wrap around the protein called the histone protein, and they're gonna turn into a nucleosome. Then the nucleosomes, they're gonna become more packed, and then they're gonna turn, when they're getting more packed, they're gonna turn to the loops, and then finally they're gonna turn into the chromatids. So the loosened packed chromatins are called as the euchromatin, but the highly condensed pack is called as the heterochromatin. So this is the euchromatin and this is the heterochromatin. So mostly chromatin is in the loosely packed in the nucleus during the interphase as they can and need to do gene expressions, and then they're gonna condense before the mitosis, which is when the cell is about to divide. Chromosomes and genetic materials. So we're gonna learn about the histories about the molecular of inheritance first. So first is Frederick Griffith experiment. Frederick Griffith was studying about the bacteria which can cause the lung inflammations. So Griffith found out that when he mixed up the heat-kill remains of the pathogenic strains with the living cell of the non-pathogenic strains, some of these living cells become pathogenic, and he called this phenomenon as transformations. So about the experiment, he tested the S cells and R cells in the mouse by injecting it in. So the S cell is the cells that gonna make the mouse die, while the R cells didn't make the mouse die. When he heat killed the S cell, mouse gonna stay healthy. But when another time he mixed the heat kill cell S cell with the living R cell, and in that time when he injected it in, the mouse died. So he can conclude in the end that this phenomenon is called as the transformations. Next is Erwin Chargaff. So, in 1947, Chargaff reported that DNA compositions varies from one species to the next. There's evidence of molecular diversity among species make our knowledge of biology develop much faster. The next person is Rosalind Franklin. So, Rosalind Franklin produced pictures of the DNA molecule using X-ray. She concluded that DNA was composed of two antiparallel sugar-phosphate backbone with nitrogenous bases paired in the molecule's interior. So, this is the pictures of one of those millions pictures that she can capture from the X-ray. The next two person is Hershey and Chase. So, in the Hershey and Chase experiment, Alfred Hershey and Martha Chase performed the experiments to show that DNA is the genetic materials by using the phage known as T2 inject into two identical bacterial cells. So, in the first one, he injects in the body, like in the proteins, where the second one, he injects the phage into the DNA, and he found out that it the bacteria that he inject in the DNA, they pass that thing that they inject to the inheritance. And next is in 1953, James Watson and Francis Crick finally discovered the double helical model for the structures of DNA. They deduct that DNA was a double helix through observations of the X-ray crystallographic image of DNA by Rosalind Franklin. Next, we're going to learn about central dogma. So it is the theory or the concept that states that genetics information flows only in one direction, from DNA to RNA to proteins. Central dogma consists of 3 steps. First is the replications. So it is the process with the genome's DNA is copied to produce 2 identical replicate of DNA from the one original DNA molecules. So in replications, the templates always be 3 prime to 5 prime, and the one that they are gonna copy is going to be from 5 prime to 3 prime. And they found out that when the DNA they like producing 2 identical replications, so they're gonna give out the original DNA to each. So 1 strands per each the daughter cells. See, this is the old strands from the original strands, and they're gonna make the new strands the copy strands. So each parent strands act as a template for building a new strands in replications. The two new daughter strands will build based on base pairing rules, which is this one. So A will always pair with T, and C will pair with G. So next is about the origins of replications. So in replications of DNA molecules, the process begins at the special sites called origins of replication, which in eukaryotic chromosomes they may have hundreds or even thousands of them. So when the double bond, the double helix, when they get separate from each other, they're gonna create the bubbles, and this one they're called as the origins of replication because it is where the replication started. Next is the leading strand's DNA polymerase can synthesize its strands continuously. Why? The lagging strand's DNA polymerase must work in the direction away from the replication fork. So as I say that the new strands, like the copy strands, they have to go from 5 prime to 3 prime. So this is the start. So in the leading strands, the start is 5 prime already, so they can continue to 3 prime. But in another one, the start is in 3 prime, but they have to start from 5 prime, right? So we have to like jump to here to make it 3 prime. 3 prime. It's from 5 prime to 3 prime. And these are all of the enzymes that helping and collaborate in replications. First is helicase. It's degrade hydrogen bonds between double helix. Next is primer. Primer produced by primase. It's used to begin building every new strands. Next is DNA polymerase 1. It's used to remove the primer and replace with DNA instead. Because of what primer is RNA, right? So they have to change it into DNA to make it the new DNA strands. Like completely, fully new DNA strands. Next is DNA polymerase 2. It's continuously synthesized by adding nucleotides onto the primer. And then having DNA ligase, it joins the new strands together. The ligase are having the important roles in the lagging strand because when you have to jump, right, you have to jump from here to producing it, meaning that there will be the gaps between like those strands. So ligase is used to join those gaps together. Next is topoisomerase topoisomerase. It stops overwinding by swiveling and rejoining DNA strands. Then we're having the last one is the single strands binding protein. It stabilizes single strands DNA until it's used as the template. DNA polymerase prove read newly made DNA and replacing any incorrect nucleotides. In mismatch repair of DNA repair, enzyme will correct the error in the base pairing. And in the eukaryotic chromosomal DNA molecules, at the end of the nucleotide sequence are the things called as the telomerase. The telomers. So it will get shorter each time of doing replications. They postpone the iterations of genes near the ends of DNA molecules. Next is codons. So codons is one of the materials in the central dogma. So codon is the series of non-overlapping three nucleotides or the triplets of bases in our genetic code. So these triplets are the smallest units of uniform length that can code for all of the amino acids. So during transcriptions, one of those two DNA strands called as the template strands provide templates for ordering the sequence of the nucleotides in an RNA transcript. But during the translations, the mRNA-based triplets are called as codons. So they're gonna call as codons in the translations. So it will be read in the 5' to 3' directions, and each codon specifies the amino acid to be placed at the corresponding positions among the polypeptide. So, what is the reason why they need 3 nucleotides per one amino acid, or one codon per one amino acid? Because in total there are 20 amino acids, but we only have 4 nucleotides, meaning that it's still enough for 20 amino acids. But what if we times 2? It's gonna equal to 16, and that's not equal for 20 as well. So we're gonna times 3, sorry. So we're gonna get 64, and that is enough for 20. So that's why we use 3 nucleotides per one codon, or per one amino acid. The gene's code is redundant but not ambiguous. No codon specifies more than one amino acid, and codons must be read in the correct reading frame, or meaning the correct groupings, in order for the specified polypeptide to be produced. Next is we're gonna first talk about the gene's expressions first. So it is the process by which DNA directions proteins synthesize includes 2 stages, which is the transcriptions and translation, which is the 2-step in central dogma other than the replications. So the experiment is George Beadle's and Edward Tennant's exposed rats mold to the X-rays, creating mutants that were unable to survive on mediums as a result of inability to synthesize certain molecules. So they use causes to identify 3 classes of arginine deficient mutants, each lacking a different enzyme necessary for the synthesized arginine. The hypothesis is first named as one gene, one enzyme, but then they restated it as one gene, one polypeptide hypothesis, since some proteins aren't enzymes, which this hypothesis states that each gene dictate productions of a specific enzyme. So next is about the transcriptions. So it is the synthesis of turning DNA into RNA or meaning the synthesis of RNA under the directions of DNA. So there's three stages. First is initiations. So the promoter signals the initiations of RNA synthesis as transcription factors bind to promoter, which is the TATA box. They call it as TATA box because the nucleotides that always stick here is always T, A, T, and A. So they call it as TATA box or TATA box. Then the RNA polymerase II bound to the promoter is called as the transcription initiations complex. Then the RNA polymerase II breaks hashbone between double helix and start RNA at the start point, like this. The next stages is elongations. So transcriptions progresses at the rate of 40 nucleotides per second as RNA polymerase untwists the double helix 10 to 20 bases at a time. RNA polymerase II will synthesize bases continuously until they found the terminated sequence. So the third one is terminations. In the eukaryotes, the polymerase continue the transcriptions after the pre-mRNA is cleaved from the growing RNA chain. But in the bacteria, the polymerase stop transcriptions at the end of the terminator. So the fourth stages is the extra stages which only have in the eukaryotes. It is the RNA processing after transcriptions. So RNA splicing is to remove intron, which is the non-coding regions, and join exon, which is the true regions that can express genes together and create the mRNA molecules with the continuous coding sequence. Alterations at each mRNA ends. So at 5' end, they will receive a modified nucleotide 5 caps, while the 3 ends get the poly A tails. See? So they're gonna cut the introns and join exon together. And then they're gonna put 5 cap and poly A tail. But in some cases, RNA splicing is carried out by the spliceosomes. So, like this picture, spliceosomes consist of the variety of proteins and several small nuclear ribonucleoproteins, and that's recognized the splice sites. Extra note is some genes can encode more than one kind of polypeptides, depends on which segments are treated as exons during RNA splicing. Such variations are called alternative RNA splicing. And because of the alternative splicing, the number of different proteins and organisms can produce is much greater than its number of genes. Next is about the materials that use in translations process. Before we're gonna know about the translations, we're gonna know about the material first. So tRNA. tRNA is a molecule that look like a clover leaf. It consists of a single RNA strand that is only about 18 nucleotide long. Each of them carries a specific amino acid on one end and anticodon on the other ends. So the anticodon base is gonna pair with the complementary codons on mRNA, while the codon base is gonna pair with the with amino acid. The correct match between the tRNA and the amino acid done by the enzymes called aminoacyl tRNA synthetase. Sorry if I pronounced wrong. Next is ribosomes. So ribosome facilitates combined tRNA anticodons with mRNA codons and protein synthesis. The two ribosomal subunit is the large and the small ones. They are mates of proteins and ribosomal RNA, which is the rRNA. The ribosomes have 3 binding sites for tRNA. The first one is the P site. It holds the tRNA that carries the growing polypeptide chain. Next is the A site. They hold the tRNA that carries the next amino acid to be added to the chain. And the last one is the E site, which is the exit site, which will discharge the tRNA and leave the ribosomes. So next is the translations process. It is the synthesis of the polypeptide which occurs under the directions of mRNA, with the help of the cells translate an mRNA message into proteins called transfer RNA, which is the tRNA. Three stages of translations. First is initiation. So first the small ribosomal subunits bind with mRNA, then tRNA joins in. Then the small subunits move along the mRNA until it's reached the start codons, which is the AUG. And then the proteins called initiation factors will bring the large subunits that completes the translations into initiations complex. Then the next stage is elongations. During this stage, amino acids are added one by one to the preceding amino acid. Each additions involve proteins called elongation factors and then occurs in three stages: codon recognitions, polypeptide bonds formations, and the translocations. And the last stages is the terminations. So terminations occurs when the stop codon and the mRNA reach the A sites of the ribosomes. The A site will accept the proteins called the release factors. Then the release factor will cause an addition of the water molecules instead of amino acid. This reaction will release the polypeptide, and then the translations assembly then come apart. Then the polypeptide chains are modified after the translation, which is the centrodogma. Yes, it is. And then during and after synthesis, the polypeptide chain spontaneously codes and folds into a three-dimensional shape. Some polypeptides are activated by enzymes that cleave them, while others comes together to form subunits from the proteins. Next is the targeting polypeptide to specific locations. So there is two populations of ribosomes, which are the free ribosomes, which is in the centrosomes, and the bound centrosome, which is attached to the ER. They are identical and they can switch between themselves as well. So the free ribosomes mostly synthesize proteins that functions in the centrosomes, while the bound cytosols make proteins that of the endomembrane system and proteins that are secretes from the cell. They are mostly free ribosomes as they finish synthesis in the centrosomes unless the polypeptide signal the ribosomes to attach the ER. Next we're gonna talk about the mutations. So is the change in the genetic materials of the cell or virus. The point materials chemical change is in the just one base pair of the genes. Only a change of a single nucleotide in a DNA template strands can lead to the productions of the abnormal protein. There is two types of point mutations. First is the base pair substitutions. It is to replace one nucleotide and its partner with another pair of nucleotides. There's three types of them. One is the silent mutations. It have no effect on the amino acid produced by the codons. Next is the missense mutations. It still codes an amino acid but not the right amino acid. While the last one is the nonsense mutations. The nonsense mutations still codes the will change the amino acid codon to the stop codons and stop the process. Next one is the insert and deletions. So it is the additions or losses of nucleotides paired in the genes. They're more serious than the substitutions. The insertions or deletions of nucleotides may alter the reading frame and produce frameshift mutations. A little note is about the mutagens, physical or chemical agents that can cause mutations. Spontaneous mutations can occur during DNA replications, recombinations, and repair, but it's gonna cause with like the small problems. So like small to like no problems at all. So next we end with the chapter 4 already, and next we're gonna start about the chapter 5. We're gonna learn about Mendel's experiment and what he knows from his experiment. So Mendel discovered the basic principle of heredity by breeding gardens pea in carefully planned experiments. So we're gonna first learn about the Mendel's experimental and quantitative approach. The advantage of the pea plants of genetic studies. There's many varieties with distinct heritable features or the characters. For example, like the flower color. And the character variants are called as the traits, such as those purple or white flower color like that is the trait. Next is mating of plants can be controlled, and each pea plants have sperms producing organs, which is the stamens, and the egg producing organ, which is the carpels. The cross pollinations can be achieved by dusting one plant with pollens from another's. So cross-pollination is the fertilizations between different plants. So Mendel chose to track only those characters that varied in an either-or manner. He also used variety that were true breeding, which is the plant that produce offspring of the same variety when they self-pollinate. In the typical experiments, Mendel made to contrast true breeding's various process called hybridizations. So a true breeding's parents are called as the P generations, and the hybrid offsprings of the P generations are called as the F1 generations. And when the F1 individual self-pollinates, the things that get produced is the F2 generations. Next, we're gonna learn about the law. We have 3 laws and together. The first one is law of segregations. The experiment is when Mendel cross-contrasting two breedings white and purple flower pea plants. All of the F1 hybrids were purple here. But when Mendel crossed the F1 hybrid, many of the F2 plants have the purple flower, but some of them have the white flower here. So Mendel discovered the ratio of about 3:1. Purple to white flower in the F2 generations. Mendel says that only if flower sorry, I'm so sorry, excuse me. So Mendel says that only the purple flower factors was affecting flower color in the F1 hybrid. He called the purple flower color a dominant trait and the white flower as the recessive trait. Yeah. So Mendel observed the same pattern of the inheritance in 6 other pea plants characters, each represented by 2 traits. What Mendel calls a heritable factor is what we are called now as the genes. So here are those other the other patterns. Am I saying right? Yes, other patterns of the inheritance that he also tried on those experiments. So this is what we're learning on. He also tried in the positions, the seed color, seed shape, the pot shape, and the pot color like that, and the stem length. So next we're gonna know about the Mendel models. So Mendel developed a hypothesis to explain about the ratio 3-to-1 inheritance patterns he observed in F2 offsprings. He found out four related concepts that make up these models. These concepts can be related to what we know now about genes and chromosomes. So the first concept is that alternative versions of genes account for variations in the inherited characters. For example, genes for flower color in the pea plant can exist in two versions, which is the purple flower and white flower. And these alternative versions of the genes are now called as the alleles. Right here. So each allele will reside at a specific locus on the specific chromosome. See? Like they have the specific place to stay. The second concept is that for each character, an organism inherits two alleles, one from the parents, which is one from mother and one from father. The two alleles at the locus may be identical as it in P generations, while two alleles at a locus might differ as it in the F1 hybrid. The third concept is that if the two alleles at the locus differ, then one, which is the dominant allele, will determine the organism's appearance, and the another one, which is the recessive allele, will have no noticeable effect on appearance. For example, F1 plants have a purple flower because the allele for that trait is dominant. Fourth concept is that the fourth concept is now known as the law of segregation. In nowadays, it states that the two alleles for the heritable character separates or segregates during gametes formations and end up in the different gametes. So gametes is egg or the sperms that get only one or two alleles that are present in the somatic cells or the organism. The possible combinations of sperms and egg can be shown using the Punnett square, a diagram for predictions the result of the genetics cross between the individuals or of known genetics makeup, which the capital letters will represent the dominant alleles, while the lowercase letters will represent as the recessive alleles. For example, here, purple with the the capital P will act as the dominant, while the lowercase p is act as the recessive, which is the white one. So we're gonna learn a bit vocab about it. Homozygous is an organism with two identical alleles for the character, which is the true breeding, while the heterozygous is an organism that has two different alleles for the genes, which is not true breeding. Next is about the phenotype and the genotype. Phenotype is the physical appearance, while the genotype is the genetic makeup. For example, PP and PP plants, the two capital one and the one capital letter and one lowercase, have the same phenotype, which is purple, but different genotype. Here, for example. Now, next we're gonna know about the test cross. If an individual have a dominant phenotype, meaning that it could be either homozygous dominant or heterozygous heterozygous. So the way that we're supposed to know is to do this test crossing. Then if any offspring displayed the recessive phenotype, that means that the individual must be heterozygous as it might have like the recessive alleles high in somewhere of like their parents. And next law is about the law of independent assortment. It states that the alleles of two genes will segregate into the other cells independent of one another. So a cross between such heterozygous the offspring is called as the monohybrid cross, while crossing two true breeding parents differing in two characters produce the hybrid. Next one is the last law is about the law of probability govern Mendelian inheritance. So when tossing a coin, the outcome of one coin has no impact on the outcome of the next toss. In the same way, alleles of one gene segregates into gametes independently of another gene's alleles. So we have two rules in this law. The first one is the multiplication rules. The rule states that probability that two or more independent events will occur together. Each gamete has a chance of carrying the dominant alleles and a chance of carrying the recessive alleles. And the additional rules is the rule that states that the probability that any one of the two or more exclusive events will occur is calculated by adding together their individual probabilities. We can apply the multiplications and additions rule to predict the outcome of cross involving multiple characters. Next, now we're gonna know more complex inheritance patterns. Because many heritable characters are not determined by only one gene but two alleles. However, the basic principle of segregations and independent assortments apply even to more complex pattern in the inheritance. So we have three types of the complex one, which is the complete dominance. It's occurs when the phenotype of the heterozygotes and the dominant homozygotes are identical. Next one is the incomplete dominance. The phenotype of the F1 hybrid is somewhere between the phenotype of the two parents varies, while the codominant is the dominant alleles that affect the phenotype and separates distinguishable way. The relations between dominance and phenotype. So for any character's dominance-recessiveness relationship of alleles depends on the level at where we're examining the phenotype. Tay-Sachs disease is the dysfunctional enzyme causing an accumulation of lipids in the brain. At the organismal level, the allele is recessive. At the biochemical level, the phenotype is completely dominant. The biochemical level, we can call it as the enzyme activity level as well. And the last one is at the molecular level, the alleles are codominant. And notes, the special note is that dominant alleles are not necessarily more common in the populations than the recessive alleles. Multiple alleles. More genes exist in populations in more than two allegic forms. For example, the four phenotype of the ABO blood group in human are determined by three alleles of the enzyme I that attach A or B carbohydrates to red blood cells, IA, IB, and I. And then the enzyme encodes by the IA alleles add to the A carbohydrates, whereas the enzyme encoded by the IB alleles add to the B carbohydrates. The enzyme encodes by the I alleles add neither. Next is about the pleiotropy. Most genes have multiple phenotypic effects. The property is called as the pleiotropy. Pleiotropy occurs when some genes influence two or more seemingly unrelated phenotypic traits. Next is epistasis. So some traits might be determined by two or more genes. In epistasis, the genes at one locus alters the phenotypic expressions of the genes at the second locus. Example, in mice and many other mammals, coat color depends on two genes. One gene determines the pigment colors, while the other genes determine whether the pig will be deposit in the hair. So we're gonna uh, tan sorry, gonna have the capital B as black and lowercase b as brown, and we're gonna have big C as color and the small c as no color. So you can see from the table from the Punnett square here. Next is about the polygenic inheritance. So the quantitative characters are those that varies in the populations among the continuum. The quantitative varies used indicates polygenic inheritance and additive efforts of two or more genes on a single phenotype. For example, the genetic inheritance is skin color in humans. The environmental impact on phenotype. So phenotype are also can get influenced by the environment as well. For example, a 100-year flower color can change with the soil's acidity. Traits influenced by many genes and the environment are called as the multifactorials. Next is about the pedigree analysis. A pedigree is a family tree that describes the interrelationship of parents and children across generations. Inheritance patterns of particular traits can be traced and described using pedigrees. Pedigrees can also be used with the multiplications and additions rules to make predictions about future offspring. Next is about the recessively inheritance disorders. So, the recessive genetic disorders appear only in the individuals with two recessive alleles. Carriers are the heterozygous individuals that have one recessive allele but show no symptoms. Next, we're gonna pick up those disorders that we can find nowadays, and like it's kind of common like that. First is albinism, which is low pupilla. Albinism is a recessive condition causing lack of pigmentation. It is a rare recessive disease which is unlikely unless both parents are carriers. Uh, a close relative marriage can increase the risk of passing on the rare recessive alleles. The next one is the cystic fibrosis. So, it is the most common lethal genetic disease in the United States. The cystic fibrosis allele results in the defective or absent chloride transport channels in plasma membrane, causing the symptoms include mucus buildup in some internal organs and abnormal absorption of nutrients in the small intestines. The next one is the sickle cell disease. So the disease is caused by the substitutions of one single amino acid in the hemoglobin proteins in the red blood cells, causing the symptoms to be the physical weakness, pain, organ damage, and even paralysis. The next one is the dominantly inheritance disorders. So some disorders can also be caused by rare dominant alleles, because it's normally caused by the recessive, right? But this one caused by the dominant alleles. It's often due to the mutations. So achondroplasia, sorry if I pronounced wrong, is a form of the dwarfism, which is one of those examples of the dominantly inherited disorders like this. Next one is the Huntington diseases. So it is an degenerative disease of the nervous system. This disease will have no obvious phenotypic effect until the individual is about 35 to 40 years old. Next is the multifactorial disorders. So this disease is like the heart disease and cancer. The disease like heart disease and cancers are result from both genetics and environmental factors, but the genetics roles is most case that is not well understood. So, like the multifactorial disorders can be found like in like those heart disease that is one of those examples, and also cancer. So the next topic is about the genetic testing and counseling. So genetic counselors help parents to know the risk of passing on genetic disorder using family histories and probability. Curious tests are available for many diseases to provide more accurate accurate risk assessment. So nowadays we have the fetal testing, which is which we have the amniocentesis, which is the liquid that baths the fetus to remove and test. And another one is the chorionic villus sampling, or the CVS. It is the sample of the placenta is removed and tested. And there's also another technique such as the ultrasounds and fetoscopy that will allow the fetal health to be known or to be seen. Another one is the newborn screening. In some genetics disorders can be detected at birth by simple tests that are now routinely performed in most hospitals in the United States. Thank you. So that's all I have for this summaries. Thank you for listening.